Thanks very much for the opportunity to speak again at the Ocular Melanoma Forum. I'm really excited to be here again. And before I introduce the panel, I give a 15-minute or so, 30,000-foot overview of what this is all about. We decided to do a little repetition. Some of the slides are presented last year, but there's lots of new people here. And repetition is king in this very difficult, complex medical and scientific area. So let me go a little bit about myself, what I'm doing, who I am. By training, I'm a biochemist. I got my graduate degrees, Master of Science and PhD degree back in Austria, where I worked on protein biochemistry. Proteins are all those compounds you heard about. PAP1 is a protein, GNAQ is a protein, so there's many different proteins. And proteins share a lot of things. I don't want to go into detail, but this is a representation of a protein. We have techniques, X-ray crystallography, where we can actually find out how they look like on a molecular level, and then we understand how they work. And in principle, we can do this for every protein, for many thousands of proteins that are out there. But I left the basic research when before I left Austria. I worked on different proteins, and then I moved to the United States in '97 and did postdoctoral research. One was at the Department of Ophthalmology. So I did signal transduction research in the eye, and I was working with many different eye models, and then I followed with another fellowship in molecular biology, medical genetics, which was much more focused on gene therapy and immunotherapy. Then I got a little tired of basic research and switched to industry, where I first did additional research in immunotherapy for a variety of cancers. The first immunotherapy actually that was approved, Provenge, was from a company up in Seattle, Dendreon Corporation, and I was in the laboratory there for 2 years, so there were many fascinating techniques. And then I switched completely from the lab to clinical research. I got certified in clinical trial management, biomedical regulatory affairs, and worked in different biotech companies. In my last position, I was the head of medical affairs for a small biotech company where we did a lot of clinical trials, expanded access programs, single patient clinical trials with investigators all over the country for different tumor types. But then I realized, in a way, in basic research, you focus on the molecules. And then in clinical research, you focus predominantly on the drug. And I decided to leave all this behind and start something completely new where I help to bridge the gap between basic research and clinical application and help patients to understand how this all works, what is out there, how to sequence the different treatment options, and most importantly, help to understand in lay terminology so that patients are able to make an informed treatment decision. And that's super important because you will see with the clinical trial panels, even for ocular melanoma, there's an increasing number of clinical trials. Which one should you choose? So there is a certain art form to help patients, and I decided to start Cancer Treatment Navigator. It's a scientific consulting company. We're a team of 5 right now. It's rapidly growing. They are all trained scientists, and I invite you to look at the website cancertreatmentnavigator.com. Okay, my short 15-minute overview. First of all, a short refresh on cancer biology, followed by treatment modalities for uveal melanoma. This will be only one slide, and it's a summary of what we already heard this morning and what we will hear later in the panel. And then I will introduce the panel, and the panel will do a fantastic job giving an overview of key clinical trials at their individual institutions. Now, I try to keep this very simple. Again, 30,000-foot view, but please feel free to interrupt me at any time if there's any questions, because it's still complex. I showed these cartoons last year, and I got good feedback because it was simple. We want to focus on only a few things and don't bring up those complex diagrams that sometimes are very confusing. So what is the difference between a normal cell and a cancer cell? the normal cell does divide as well. For example, our bone marrow is in a constant state of dividing and producing new cells, our blood cells, and that's a necessary process. Cancer cells divide as well. Now, the way how every cell works in cell division is a simple process called cell signaling or signal transduction. And in principle, it works that there is a cell receptor, and you see this on the very top of this pink cell, of the large cell. The cell receptor is nothing else than an antenna, and the antenna can speak with the environment around the cell. And then there is signal molecules, and I only highlighted 3, without a name, just different shapes and different forms. And what happens if a growth factor attaches to the cell receptor, and I simply call it just growth factor, this triggers a cascade of many different reactions. And we call this the cell signaling or signal transduction, because the binding of a growth factor on the cell triggers an electrochemical signal that switches on signal molecule 1, which triggers an electrochemical signal that switches on signal molecule 2, and so on and so forth. The final end result is that the cell nucleus, which houses the machinery where all the genes that are nothing more than complex recipes for products are started, and the cell can either divide or produce something. So that's going on in a normal cell. Now, what's the difference in a cancer cell? And one of the major hallmark differences is that there are mutated proteins. A mutated protein, again, I grew up in Austria, so the German word for protein is Eiweiß. That means egg white. So that's because egg white is mostly protein. And the protein is a product of a gene. So it's very important to see the difference between a gene is nothing more than a recipe for the product. It's like a gene is a page in a big book. And the book is a chromosome. And if you open up the book, there's a page, and the page is the gene. On the gene, you find a list of 10 ingredients to bake a cake, the product. It's an analogy of what a gene is. A gene is a very complex recipe. It might be multiple pages with multiple instructions on how to make the product. For example, egg white or BAP1. Now, the machinery to house the gene can get mutated. And the resulting protein is different. And that's a hallmark in cancer. We have multiple mutations in cancer. While the normal cell relies usually on a growth factor to trigger the signal and tell the cell to do something, the cancer cell can work independently of the environment. And the mutated protein can start the electrochemical signal on its own, simply speaking. And again, those are very simplified cartoons. And then the signal is transduced into the cell cycle automatically, ongoing, without any stop. And this leads to cell division on a much, much faster pace, and a tumor can arise out of this. Now, mutations are not happening on the protein. The mutations are happening in the recipe, in the genes. And we can change the genes so that we can change the order of which the different ingredients are put together to form a protein. And the way how those changes happening is radiation. UV radiation, for example, in melanoma, we know plays a role. The cell division, how the DNA, how the big books of life are copied can be faulty. That can lead to mutations. Viruses and other infections can cause mutations. And a lot of environmental chemicals, unfortunately, can cause mutations as well. Again, all this leads to different mutations, and the goal in targeted medicine is to learn about the mutation, know where they are in the book of life, the chromosomes, the genes, and develop medicines for each unique mutation. And that's the new treatment paradigm of targeted therapies, which is unlike chemotherapy. Chemotherapy usually targets every dividing cell. Chemotherapy is very good in destroying and stopping cell division. Unfortunately, it's also very good in destroying and stopping cell division in a healthy cell. That's why we have usually a fairly high rate of side effects. Now, going back to targeted medicine, in skin melanoma, also called cutaneous melanoma, we have one example for a mutated protein, and I just pick one. It's called BRAF. It's not important what the name of this molecule is, but it's a molecule that is mutated in quite a high number of cutaneous melanoma. And again, the signal starts from the mutated protein and forms a tumor much more efficiently as if it's not mutated. Now, targeted medicine, again, targets a specific mutation. And this is an example in cutaneous melanoma where multiple medications have been developed. For cutaneous melanoma, one of them is called dabrafenib. The other one is called vemurafenib. So those are approved products for cutaneous melanoma. And the outcome is fantastic. In many patients, they have a great clinical benefit of shrinking the tumor. It's also given in combination with another medication that targets another gene mutation. Now, in uveal melanoma, we don't usually have, or extremely rarely, I don't know of any case, a BRAF mutation. In the uveal melanoma, class 2 melanomas, the vast majority of patients have a mutation in another protein called GNAQ or GNA11. They are interchangeable. Let's call them almost identical. I don't want to go into the detail what the differences are. And what can we do? Can we do the same what we do with BRAF in cutaneous melanoma? Unfortunately, not yet, because we don't know how to stop the mutated protein. So we have big question marks. And what I showed you in the very beginning, this is a representation of how the GNAQ looks like, or the GNA11, again, identical almost. And it's a very complex way. What currently is going on, and I'm involved in a research project with Dr. John Sondek at the University of North Carolina, Chapel Hill, who looks into thousands, hundred thousands, millions of compounds that could fit into this molecule that is mutated to make it stop. I was lucky to be involved because one of the patients in the room here is involved in this project, and it's a fascinating project that not only looks at how can we stop GNAQ that is mutated in uveal melanoma, it also looks, are there other mutations that are very similar, and how can we stop the other mutations? In the meantime, the way how we design clinical trials, there's first a scientific hypothesis, and then the scientific hypothesis is translated into clinical trials, and we will hear from the panels. So one hypothesis is that this molecule that is mutated and constantly fires a signal actually is not that simple that it's one linear signal. Usually it branches out, and I showed the branch here in two different directions, where two different branches fire signals and tell the cell to form a tumor. Now, what one clinical trial that will be presented by the panel today does, it targets so-called downstream molecules. So if it's constantly firing, why don't we target those molecules that are further downstream? And there's one great clinical trial. We don't have any data yet how it works, but there's one trial that targets a molecule called PKC and another molecule called PI3K. And in combination, the hypothesis is to stop the signal and stop the tumor, help shrink the tumor. And this is currently in clinical testing. And the panel will hopefully talk about this trial. In theory, it's all more complex. It's not that easy because this is the more complex pathway. And here again, it's the same what I showed you before, the mutated GNAQ or GNA11. And we understand what it does. And you see the two main branches of the signaling pathway. And those are currently molecules that we have to interact with other so-called downstream molecules because they are downstream of the mutations, like a river flowing downstream. If we can't dam the river on the very top, why don't we try to dam the river further downstream? And that's going on again with focusing on PKC or PI3K. And this is just an example. Now, I don't want to discourage you. The picture is much more complex. This is a more integrated cancer pathway. And if you think this is too complex, look at this one. This is, as we know, the integrated breast cancer pathway. You find this in databases. And in fact, what you see here, green is accelerating something, red is breaking something. It is very complex. Now you might think, oh my God, how can we ever find the right mutation? How can we find the needle in the haystack? We can, as this example showed. And that's what we want to achieve in ocular melanoma as well. So we found one pathway that is very, very delineated, and we can stop it, at least for a reasonable amount of time. Now, there is, of course, complexity because some patients recur after this. Well, because another pathway might be upregulated, and cancer is constantly developing. There's mutations going on, and different clones are arising, and heterogeneity of the cancer is another topic that I don't want to go even into detail. But again, don't get discouraged if you see something like this. This is more the reality, and this is only a snippet. That's what we know currently as scientists. But we are successful, and the reason why we do clinical trials is to find those molecules, to prove that it works, and to find new therapies that help patients. Now, before I get over to the panel, the treatment modalities that will be shown again by three categories: by stage, by location, and by type. And by stage, I mean, are we in the adjuvant setting? So you only had a tumor in your eye, you had the primary treatment, and the adjuvant treatment is the setting that helps to avoid metastases from ever developing. Or the metastatic setting. If you already have metastases, how can we stop them? How can we halt them to provide clinical benefit? By location, we have eye-directed therapy, liver-directed therapies, and systemic therapies. And then by type: surgery, radiation, chemotherapy, targeted therapy I was talking about, immunotherapy, we will be talking right after the panel, and combination therapies. Two more slides before I introduce the panel. Last year, it was interesting, I looked at clinicaltrials.gov and I typed in ocular melanoma and uveal melanoma. We were talking about, there's a difference. For the sake of the Ocular Melanoma Foundation, we call it ocular melanoma. Scientists and clinicians make a differentiation between uveal melanoma because ocular melanoma includes other cancers that are not really the topic of this gathering. But again, if you look at ocular melanoma, we had last year 78 studies that are open, and 34 studies were found for uveal melanoma. This year, and I searched this yesterday on the plane, it's 86 studies and 41, so a little bit up, which is a good sign. However, it's not enough. If you search skin melanoma, you find more than 500 trials that are currently open. And that's something why the Ocular Melanoma Foundation is so important, because to create a voice for cancers that are rare. Ocular uveal melanoma is a rare form of cancer, a rare form of melanoma that is different than skin melanoma, as you all know. And the stronger the voice, the more likely to secure additional trials and force industry to get interested in clinical trials.